Oh, right. So without further ado, I want to take this opportunity to introduce our keynote speaker, Dr. Jens Juul Holst. He is a professor of medical physiology and deputy head of the Department of Biomedical Sciences, University of Copenhagen. He is also research director at the Novo Nordisk Foundation Center for Basic Metabolic Research at the University of Copenhagen. Professor Holst's scientific work has been focused on the regulatory peptides of the pancreas and the gut and their importance in the regulation of the functions of the GI tract and metabolism, with particular focus on blood glucose and appetite regulation, obesity, and diabetes. I think I just gave his keynote presentation for you. There are many, many other things that he is known for, but without further ado, I'm going to take this opportunity to say, please join me in welcoming Dr. Holst.
My presentation disappeared. I hope it will come back. Let's see. Come on. Yeah, what we need. So here we go. So thank you very much for inviting me, and now that it seems to work, let's get started with the presentation here, which is about mechanisms in bariatric surgery. And what I'll be talking about is particularly the gut hormones and their possible effects on diabetes resolution and weight loss. And so we got involved in this. Now it's quite a number of years ago, 2004, 2005, when we were approached by people from Harvard who had these patients with severe hypoglycemia after gastric bypass operations. And we got some samples and we measured them for the content of GLP-1, which was a hormone that was discovered in my laboratory many years ago. And we found these extremely high levels of, as you can see, 300 to 500 picomoles per liter. And that was really completely absurd high levels. So clearly that was something very, very interesting to look at here. We went on to collaborate with Judy Corner, and we did a study where we looked at postprandial levels in a larger group of patients with banding or bypass operations. And as you can see, the GLP-1 levels were also, in this case, very, very high, whereas in banding and in patients, control patients, there was not nearly a similar elevation of the GLP-1 levels. This doesn't, here we go. Wonderful. So we went on to collaborate further with the people at Harvard, and this time looked at people with neuroglycopenia after gastric bypass surgery. And as you can see, they have these low levels of glucose here, and it was clearly hyperinsulinemic what went on. So it was interesting to measure. I have a problem with this. So it was interesting to measure the GLP-1 levels, and again you can see that these are sky high in these people, and we got the impression that this was really something to go for. It was very likely, given that GLP-1 is an insulinotropic peptide, that it had something to do with the hypoglycemia, but certainly also with the remission of diabetes in the other patients. So what we have is now a schedule, a mechanism for what happens after bypass operation. And the important thing that we have here is that after the operation, immediately after the operation, we have this tremendous perioperative calorie restriction. And that causes a doubling of hepatic insulin sensitivity. That is a very important parameter. The next thing that happens is, as soon as the patient starts to eat, is, of course, that we have this unretarded passage. Unretarded, there's no restriction here. Unretarded passage of nutrients into the small intestine and increased absorption, and I'll go a little bit into that. We have this abnormal exposure of the distal small intestine with abnormal secretion of hormones like GLP-1 and PYY. And we can then analyze what that means in terms of insulin secretion and also appetite, as I'll show you. Eventually, with time, we have a weight loss. And that also is extremely important because that causes a further improvement of now peripheral insulin sensitivity. And that alone can, of course, induce diabetes resolution. So these are the main factors that we will be looking at. Now, here we have the first stuff, which is the perioperative improvement in the calorie restriction and the improvement in hepatic insulin resistance. And you can see here that we have the type 2s up here with a very high, this is normal glucose tolerant, this is type 2s with a very high insulin resistance measured by HOMA-IR. And as you can see, 1 week after the operation, we are down to about 50% of what it was. And note that we are now down to the levels of the healthy controls. So this is really an amazing reduction. And as you can see here, until 1 year, they continue to drop, to improve. That's HOMA, you say, and perhaps you don't like HOMA because it's a primitive way of doing it. But we also took the effort to look at hepatic glucose production with clamps and did this. And here we are measuring the basal hepatic glucose production. As you know, diabetes is an increased basal glucose production. And sure enough, after 1 week, it is down to exactly the levels that we find in the controls, as you can see here. That one does not decrease a lot further anymore, but as you can see here, we have the hepatic insulin sensitivity, which increases and increases just as I showed you a minute ago. The peripheral glucose disposal, as measured with the clamp technique, does improve, but only after 3 months, and that is in parallel with a major reduction in body weight. That's when you start to lose weight, also in the muscles. So is that important? Here is a study from Roy Taylor. He's from Newcastle. He's the one who thinks that you can cure all diseases by not eating at all. But here he has done a study where he's looking at very low-calorie diet and Roux-en-Y gastric bypass and has actually quantified the hepatic fat content after a week with MRI technology. And he found that fat mass really decreased in Roux-en-Y and diet groups, but the reduction in liver fat was greater after Roux-en-Y than it was in the diet group. And it was a lot. As you can see, there's a reduction of 30% of the fat in the liver after 1 week. And that really is a major difference. So does that matter? And here's a beautiful study that has just appeared in Diabetologia, and it shows the correlation between HOMA-IR, the parameter that we just talked about, and liver fat measured by MRI. And you can see that there is a very strong and very significant correlation between liver fat and HOMA-IR. And that means that you can actually use the HOMA-IR as a measure of liver fat, which is extremely useful, I think. Now, the next is, of course, that we have the unretarded passage, and I'll show you that. Here we have it. And one of the best ways, I think, to measure this is really to use a liquid meal and look at paracetamol absorption, and that's what we've done here. And here you see, before the operation, the normal paracetamol absorption curve, and then after the operation. And there can't be any doubt here that this hugely accelerated entry of nutrients into the small intestine has a lot to do with what happens. You can see that the highest point is immediately after the food intake. It's really a major event here. Now let's have a little bit more in detail about what happens to glucose. So for this, we have looked at some people, these were with normal glucose tolerance, and we gave them a mixed meal test, and that contained tracers. So this is a triple tracer technology where we can look at what happens with the glucose in all the tissues and the fluxes that we have. So first of all, let's have a look at plasma glucose. So this is a typical pattern before and after operation. You can see the increase in glucose, beautiful steep increase, and the fall after some time. Very typical of our patients. So what happens actually? So here we have the total rate of appearance of glucose. This is all the glucose that comes into the circulation from whatever source, kidneys, liver, and food, of course. So this is the total. You can see this tremendous increase after the operation. So the glucose comes into the circulation in enormous amounts. So here we have the endogenous production in the liver of the glucose, and you can see that there is no real difference between the preoperative and the postoperative findings here. So it's not the liver, it's shut off anyway. So here we have the rate of appearance of all glucose, and again you can see this tremendous rise after the operation. So it is indeed the meal-related glucose that enters the circulation, and also it enters all of it. The recovery is very, very high. And then, eventually, all the glucose is also put into the tissues. This is the rate of disposal of glucose into the tissues, and that is also extremely accelerated. And as I'll show you later, that is a function of the insulin increase, of course. So that's what explains this glucose curve. So in summary, what we have is that we have this tremendous change in glucose profile after the operation, and we have the same inhibition of endogenous glucose production. We have this very rapid rate of absorption of glucose and a high recovery, which means that we don't lose anything. So those that think that you can invent some of this by changing the combustion of glucose in the gut or changing the production of glucose in the gut, they can forget it, because here we have a complete account of what goes on. And then eventually, with the insulin secretion, we have also this increase in glucose disposal. So now we've accounted for exactly what happens with the glucose. Here comes the surprising finding. We also did this with proteins. So now we're looking at protein at the same time. And this protein meal was labeled so that the protein had a built-in radioactive leucine, for instance, in it. And here we're looking again at these things. So here, first of all, we have the profile, the plasma profile of the leucine. And then here we have the rate of appearance. No, that's the endogenous production of leucine, which goes down as you take the meal in. So that's not the reason for, that's not the explanation of this profile. And here, then we have the rate of disappearance. It's taken up very rapidly in the tissues also. But first of all, it is the ingested protein that is taken up extremely rapidly. And this means that it must reach the common limb after the operation extremely rapidly, and that there must be sufficient amounts of enzymes in the common limb to split the proteins and ensure an extremely rapid absorption of the amino acids. So that's what happens here. So that's why I've emphasized that this actually even occurs in the common limb. So those that think that everything goes on in the actually root limb, they are wrong. It's in the common limb we have the exciting events going on. Now, let's move on and have a look at what happens in the gut. That's the hormone secretion. And here we have all these hormones that are interesting: the ghrelin, the GIP, the CCK, and the GLP-1 and the PYY. And what happens after the operation is, of course, that this is shifted around so that now foodstuff enters at different sites. And that's one of the things. So what happens with GIP, the glucose-stimulating hormone, the insulinotropic peptide? Nothing much. CCK repeatedly shows increased secretion after the operation, which is interesting. And I do think that CCK may be part of the equation. We have the tremendous rise in GLP-1 secretion, and we have a tremendous rise in PYY secretion. And those are some of the main figures. Let's have a look at the GLP-1 secretion again. So here we have it before the operation, and 1 week after, and 3 months after, and 1 year after. Tremendous rise, up to 30-fold increases in the secretion in both type 2s and normal glucose-tolerant individuals. Not only does GLP-1 increase, but the proglucagon molecule here is broken down in the gut to also produce glycendin, which is this molecule, and oxyntomodulin, which is this molecule, and GLP-2, which is this molecule over here. And all of these also come out in the circulation. And this is shown here for glycendin and oxyntomodulin. Oxyntomodulin is not interesting in healthy individuals. The concentrations are too low. Glycendin, we don't know much about it, but after the operation, you have again tremendous rise in glycendin and oxyntomodulin. And since oxyntomodulin also has appetite-regulating properties, this may be interesting. We'll come back to that. Are there differences in the various operations with respect to these patterns? Yes, of course there is. If the things are tied together the way I've described it. And here we have an experiment with a long-legged Roux-en-Y gastric and a short one, a classical one. And you can see that those with the long legs, they have larger GLP-1 responses. So this is one way of manipulating what goes on. You can do things with that way. So which of the foodstuff that we take in is the most important stimulator of these hormones? And that we've looked at here in a study where we looked at a carbohydrate meal with a large amount of dextrose, and a fat meal with a large amount of lipid, and a protein meal with a lot of whey protein, and they are caloric, these meals, and they have the same volume. So we gave it to our patients in a soup so that they could drink it easily, and then we looked at what happened to the hormones. So now here we have O1Y, and here we are looking at fat and protein, and here we have the controls. Well, there is an increase after O1Y, but it's not quite what I showed you before, is it? But let's have a look at what happens with glucose. Here we have it. So it's the glucose, it's the carbohydrates that cause this tremendous rise in GLP-1. And that really means that we must concentrate on that mechanism to understand this. But what is interesting is that it differs for the other hormones. So here you have O1Y with PYY, and you can see again glucose is the one that drives even the PYY response, whereas in the controls it's actually the fat content that drives the responses. And here you have GIP. As I told you already, GIP doesn't change much. Glucose is good, but in the controls, it's actually the fat here that is the greatest stimulus. So these things are important, and that means that the hormone responses will depend somewhat on the composition of the meals. CCK, again, shows increases with glucose after the operation, but certainly with fat before the operation. So again, a marked difference. Here's one more hormone for you. And that's a hormone that people think also participates in the regulation of appetite and food intake. Neurotensin, it's called. And neurotensin particularly is elevated after the fat meal. So there's a mechanism for each of these hormones, and it's different. It depends on exactly what the stimulus is. And neurotensin may be particularly involved in fat-induced satiety. Why do we get these exaggerated responses? I said it was derived from absorption, incredible rapid exposure and absorption of the nutrients. But are intestinal adaptations and mucosal hyperplasia essential mechanisms here, as it's been said by several groups? Well, I'd like to return to this old case story we had, where a patient had a O1Y gastric bypass. He had a BMI of 50, and he was on insulin and oral agents because of diabetes. And then he had a gastric tube inserted because they felt that something was wrong. There wasn't really something wrong, but nevertheless, he was after 5 weeks, he was dismissed. But then at that day, we had the opportunity to study this guy with a meal that was either introduced via the gastrostomy catheter or via the mouth. So that's what we did and what happened. So here we have the glucoses, and this is via the bypass. And you can see that there's a rapid return of the glucose values, as I've shown you already. Whereas when he was fed via the stomach, the old route, he was still a diabetic. So on 2 consecutive days, he shifted from being a diabetic and a non-diabetic. So that cannot be associated with a major structural change in the gut. And sure enough, if you look at insulin and GLP-1, these were the responses. This was the bypass meal. This was the stomach meal here. And you can see that their relationship is so that the GLP-1 response comes immediately before the insulin response, as it should. We have another example here. This was the patient that suffered from severe hypoglycemia after bariatric surgery, and you can see it here. And then he was operated by a surgical reversal. This was difficult, but it was possible. And so what happened before the operation was, of course, here that we have this tremendous insulin rises here, and we have the GLP-1 rises, but after the operation, the hypoglycemia was away, the insulin was away, the GLP-1 was away. Interestingly here, there was also an immediate change in ghrelin, and ghrelin may be another of the factors that are important for appetite regulation. And sure enough, as the ghrelin increased, the patient also increased his food intake, and he is now weighing more after this reversal of the operation than he did before. Now we will look at insulin secretion with GLP-1. And here we have the insulins of the group that I showed you already, those that were followed at 1 week, 3 months, and 1 year. And you can see the change in insulin secretion, as we've seen a couple of times now, with this early left shift and rise in insulin secretion. And actually, if you look at it, the new response, insulin response, looks very much like that in the normal glucose-tolerant individuals. In other words, in a way, you have normalized the insulin response. So we looked at them, and the way we could do this, investigate the importance of GLP-1, is to use the GLP-1 receptor antagonist exendin 9 to 39. And that's what we did. And here we have them, the experiment before, 1 week after, and 3 months after. And took a lot of samples here, and here we have them. First of all, glucose tolerance. So these were the diabetics before. This was 1 week after, nice improvement. 3 months after, even better improvement, the A1Cs down here. And you can see 2-hour value, 10, 8, 6.5. Isn't it a beautiful result? The diabetes is gone completely. And then let's give the patient the GLP-1 receptor antagonist, and what happens? You impair glucose tolerance immediately, both before and after, and 3 months after. And if you look at the area down here of glucose, the AUC, you'll see that after the antagonist, we are back to completely what it was before the operation. In other words, all that was gained by the operation was lost when we gave the GLP-1 receptor antagonist. And the same is true for insulin secretion. Here we have insulin secretion rates, and here we have with Exendin, and you can see the decrease before the operation, but certainly an important decrease in insulin secretion after the operation. And again, if you look at the area under the curve here, you can see that it's the same as it was before the operation. In other words, all that was gained by the operation in terms of insulin secretion was lost after the GLP-1 receptor antagonist. The real important parameter here is beta cell glucose sensitivity, because that's what GLP-1 does. It increases the beta cell sensitivity to glucose so that it produces more insulin if glucose rises. And how can you determine that? You can do that by looking at insulin secretion rate, as we did in these meals here, and the glucose concentration profile. And then you can cross-correlate those two here. Here you have them, and here you have them, the straight line that usually comes up here. And that tells you how much insulin is produced. The slope of this line tells you how much insulin is produced per increase in glucose unit. That's the slope. We have it here. That is the beta cell glucose sensitivity. And that we calculated also here. And so we have them here. And again, you can see that the beta cell sensitivity to glucose is tremendously increased by this GLP-1 rise. And after the antagonist, all of it is lost. So is GLP-1 a good candidate for this? Some people think that it is not. I think it's a very good candidate, and here's some of the evidence. Why is it such a good candidate? Have a look at what happens with the GLP-1 agonist if you go out and treat people. So these are treated for 2 years, 3,000 of them, and have a look at the A1C levels, and have a look at the body weight versus semaglutide. Beautiful result. So it can do it. It can do it. There's no doubt about it. And actually, in a recent obesity study with semaglutide, you may have seen it, there was a weight loss of 13.8%, corresponding in some patients to 20 kilograms in average to the semaglutide treatment. So it can do it. Was there any adaptation of beta cell function? We looked at that by clamping here, and then measuring insulin secretion rates to the clamp and arginine here. And we did that before and 3 months after and 3 years after now. And you can see that there's no real change. And here we're looking at it again before. This is insulin secretion. And after, and these are controls. And if anything, there's a little bit of a decrease in beta cell responses here, but no increase, no change. So it is not changes in the beta cell mass. It's not changes in the intrinsic beta cell function that does any of this. And here you have the GLP-1 responses after 3 years, the GIPs and the glucagons. I will not talk about glucagons because it's so terribly complicated. I'd love to do it on another occasion where we have more time. So now let's have a look at the weight loss, because as I said, most of this is really driven by weight losses, both the early improvement in insulin sensitivity and also the late improvement in insulin sensitivity. So how do we get this weight loss? Well, we have this exaggerated secretion of GLP-1 and PYY, also some of the other hormones, as I told you, but let's now analyze what happens with GLP-1 and PYY. So here we have some experiments we did with infusion of GLP-1 and PYY to people, and we looked at food intake. This was a low-dose infusion of GLP-1. It didn't really do a lot of PYY here. It did a little more maybe, but the combination had a potentiating effect on energy intake. Very strong effect indeed. So once you have both of them, you can expect a bit more than the sum of their activity, which is shown here. So can we use this extended 9 approach to analyze the effect of GLP-1 on food intake? Yes, of course. Let's try. So here is before, these are patients that are about to be operated. This was before the operation here, and we're looking at energy intake after the antagonist. It increases, as we would expect, if this is to play a role in the regulation. Now let's look at after the operation. Oops, no change. Not so good. So it doesn't work, or does it? So the problem is, if you look at the concentrations of GLP-1 before the operation, here we have them before the operation, and after the operation we have the usual increase. If we give extendin 9 as we did, you note that the concentrations increase. And that's because there is a negative feedback regulation of GLP-1 on the L cell. So that's quite a sizable increase. And after the operation, it's even bigger. Now, PYY comes from the same cell. Is that also regulated by the same feedback mechanism? Yes, it is. So this is before, and this is after extendin 9, and it increases. And this is after the operation, and this is after extendin 9. Now there is a huge increase in the secretion of PYY. So as we do this experiment, we get an increase in PYY instead. And that's why we don't change anything. Can we prove that? So the point is here that if you want to study this process, you need to be able to block both of the hormones, not one alone. And for GLP-1, we have this nice receptor antagonist. For PYY, there is no antagonist. But there is something else. PYY is formed as the hormone PYY1 to 36, and it must be degraded by dipeptidyl peptidase 4, the enzyme, to PYY3 to 36. Then it acts on appetite and food intake. And that we can block with a DPP-4 inhibitor, and we have one. There are many. This is sitagliptin that we used for this. So now we can block both of the hormones. So now let's have a look at what happens. We did a study with a meal test here, and they had a meal here, and they had an ad libitum meal afterwards, and we could do that with a placebo where we had both the hormones responses, GLP-1 and PYY. We could use extendin 9 and take away the GLP-1 component. We could do a DPP-4 inhibitor and take away the PYY, and finally we could take both of them away with a combination of those two. And that's what we did. So let's have a look at what happened. So first of all, we have the concentrations of GLP-1. And for this, I'm showing you the intact, the active hormone, which is the one that acts on appetite here. And so let's have a look at what happens. So this is the placebo, and all of it is after the operation, note. And then we give them extendin 9, and we have the rise that I told you about. And then we give them a DPP-4 inhibitor. That DPP-4 inhibitor also increases GLP-1 as it should. And then we give them a combination, and we get a tremendous high response of intact GLP-1. This is really, really, really high. Now let's look at PYY. So here we have the placebo experiments. This is the PYY response. Now let's try to see what happens with extendin 9. We get a huge increase. So as I told you, if you block with extendin 9, we get this tremendous rise in PYY. Now let's see what happens after the DPP-4 inhibitor. And this was the placebo, and now we have the DPP-4 inhibitor, and everything is gone. So it was possible. It was possible to block PYY. And here we have it then in the combination. We couldn't block it completely, but it was down very, very much to a small percentage of what it was. And the consequences for food intake are here. So this is placebo, this was extendin alone, this was DPP-4 inhibitor, and this then was with the combination of the two, and then we had an increase in ad libitum food intake by 20%. So indeed, if you blocked both of the hormones, it was possible to demonstrate this. We've done some recent studies to see if these combinations of hormones are good, and here we have included GLP-1, oxyntomodulin, the one I talked about, and PYY, and looked at energy intake and energy expenditure in obese and non-diabetic volunteers, And here we have them with their food intake, and you can see that it's possible on all meals to block their food intake quite substantially with this infusion of these 3 hormones together. I heard a presentation recently by Michael Schwartz where he said that all of it was about glucose effectiveness. And that can fortunately, that was in mice, of course, but that can fortunately be studied in people, because all you have to do is a frequently sampled intravenous glucose tolerance test to estimate first phase insulin, insulin sensitivity, and glucose effectiveness. That's how you do it. It's defined this way. So let's see what happens. So the results was that the first phase insulin secretion increased 2-fold. That was before and after 1Y. Insulin sensitivity increased at week 1, as I showed you already, and at 3 months in the peripheral tissues, again, as I showed you, and glucose effectiveness didn't change at all. So glucose effectiveness does not change after 1Y gastric bypass. What about energy metabolism? In all the animals, in all the rats and all the mice that have been looked at, energy expenditure increases after these operations. What about people? So here we are looking at it in a smart way, because these people, they are looked at here, the screen, they have the baseline, they're put in a respiratory chamber, and then they have the operation, or they are followed for a period where they are on exactly the same energy restriction as these guys here, and then the other group has their surgery here. And that way we can compare exactly what happens and titrate it precisely as to energy intake during this duration here. So that's what we did. A lot of things was measured, and it's all appeared in this actually very nice, I think, by Smith here in 2016 in IGO. And it's a long and complicated study, and I recommend it very much. Julia was very, very good at it. Here we have the respiratory chambers, and here we are showing that they're in a similar and constant negative energy balance. And these are then the recordings from the respiratory chamber, and you can see that they fall after the operation and nothing else. You can judge all of the food intake, the energy intake increases, the diet-induced thermogenesis here, and all the nightly responses. Everything can be accounted for, and what we can conclude about this was that energy expenditure decreases. So the acute weight loss after 1Y is not mediated by increases or preservations of energy intake, and their appetite was decreased, and we found that this could, by statistical analysis, be explained by GLP-1, PYY, and ghrelin came in as an interesting thing. Now, finally, what about sleeve gastrectomy? Is there something we can learn here? And we have now engaged in a study comparing sleeve versus 1Y. And for this study, we've done a cross-sectional study in weight stable, 12 months, about 12 months after the operation, 12 here, 12 here, 12 controls. But they're matched individually for original BMI, for sex, for age, for BMI lost by the operation. So this is not a comparison of what happens with Roux-en-Y compared to sleeve, not at all. It is a comparison of those that had exactly the same weight loss at the same time. So this is a very specific population. And the time from surgery, as I said here. So let's have a look at it. They had a liquid test meal here again, and we took a lot of blood samples. And here you can see that they had the weight loss, the excess BMI lost was the same, and the A1Cs were the same, the weights were the same. And here we have the glucoses. So these are the controls. These are the ones with the sleeve, and these are the ones with the 1Y. And the sleeves put themselves right in the middle between the others. Here we have the insulin secretion rates. So, controls, sleeve, 1Y. Again, they put themselves in the middle. And beta cell function. Now, this is indicated as the disposition function. That is insulin secretion corrected for insulin sensitivity. And here we have it. Tremendous increase in disposition index in the 2 operated groups here compared to the control group. And all of it is really due to differences in HOMA, as you can see, because the actual insulin secretion didn't change that much. So it's the insulin sensitivity that distinguishes these 2 groups. And then paracetamol. So what is the actual rate of entry into the small intestine? And this is Roux-en-Y, as this is sleeve, and it's not nearly as fast as it is after Roux-en-Y in the sleeves. And sure enough, the GLP-1s are also much higher in Roux-en-Y than they are in the sleeves. GIP doesn't really give us a clue. But here, the only thing that comes out as a very interesting parameter here are ghrelin levels. This is active ghrelin, and here you have the controls, and here you have the decrease in the Roux-en-Y, and here you have the decrease in the sleeves. And at present, it's the only explanation I have about the differences between the two groups. That's ghrelin. So, my conclusions: the rapid entry and absorption of nutrients into the small intestine causes an exaggerated release of several gut hormones, including GLP-1, neurotensin, PYY. GLP-1 and glucose are responsible for the increase in insulin secretion. GLP-1 and PYY act in concert to inhibit appetite and decrease food intake after Roux-en-Y surgery. CCK, ghrelin, and NT may also play a role. I think they do. Insulin function varies very little over time. Energy expenditure increases. Glucose effectiveness is unchanged. And sleeve gastrectomy may actually be somewhat different. And here, perhaps, it's the ghrelin, the active ghrelin levels that really distinguish between the two operations. So that's as far as we've come today. Thank you very much.
I'm going to open this up to anybody who may have any comments or questions for Dr. Holst. The microphones are in the back, in the middle there. Go ahead.
Christina Rother, NIH. I'm back here on the other side, back here.
Oh, yeah.
So this was a beautiful talk. And ultimately, do we need to conclude that ghrelin is weaker than GLP-1 because people after gastric bypass lose more weight than
So
after?
one of the reasons that we have been reluctant to look too much at ghrelin is the fact that it changes the ghrelin responses. So the decrease we have in the beginning at 3 months, or 1 week at 3 months, and about a half year, it goes away. And after a year, a full year, we've published that, the ghrelin levels seem to come back. And under those circumstances, we're not too happy about putting too much weight on ghrelin. But I must say that the recent data we have here with the sleeves, they really bring back the enthusiasm for ghrelin, and I think we have to do a lot more about this in the future.
May I ask a very quick second question, and that is, when we look at the 0 and 120-minute glucose concentrations, we talk about resolution of diabetes. But you have beautifully shown that people, despite having normoglycemia at 2 hours, consistently have these very high excursions at 60 minutes.
Yeah.
What does this do to metabolism, to the brain, and so on in the long run, cardiovascular disease?
Yeah, so in fact, it doesn't really come as a surprise. I trained in surgery when I was young, in GI surgery, and we knew that whenever we did something with the stomach, we would have these we call them oxyglucosemia or oxyglycemia, to indicate that we had very high rises and very rapid decreases. I think what matters the most is that the A1C levels come down so very nicely. So even if you have these high peaks, which are quite strange and quite abnormal, I'll have to say, but they eventually don't mean too much for the average glucose regulation of the patient, and the A1Cs come down, and as you know from wonderful studies, particularly from the SOS studies, there are great improvements in diabetic complications in these patients and prevention of diabetes also. So regardless of the fact that we have these high rises after the meals in the beginning, it really means that it is getting better.
I wanted to thank you for that exquisite talk. It was excellent. I have one question that you kind of alluded to in your preliminary remarks. about the root limb and the common limb. Basically, just a comment on the data by Dr. Stropoulos. I think that his animal study, and it's currently being translated into a human study, actually, at our center. And I'd just like you to comment on how essentially he can sort of account for about 70% or so of the improved glucose metabolism by reprogramming the metabolism in the root limb. So I wanted to get your perspective as to how does this fit, so to speak.
So I'm not quite sure I understood all of it, but are we talking about the importance of glucagon in this business? Is that the main thing?
But essentially, you're talking about the improvement in glucose metabolism, basically clinical insulin are measured by hemoglobin A1C, however you want to measure the improvement. But in other words, I guess there's some incongruence, so I'm trying, and it's a very complex question, as you elaborately displayed in multiple ways. But I guess I'm trying to fit into my mind. I'm not exactly a basic scientist, and I'm trying to fit some of the puzzle together.
Yeah, I can understand. There are many difficult things here. But I hope that it is about glucagon because I know something about that. And sure enough, I've shown you that the glucagons are increased after the operation, and surprisingly so. And we've come to the conclusion that the glucagon after Roux-en-Y comes from the gut. It is really glucagon. We know that for sure. We've done mass spectrometry to identify it. We've found the cells. We know that they can produce glucagon. And we have compared them to people with total gastrectomies, with total pancreatectomies, where the reconstruction after the operation sometimes resembles very much a Roux-en-Y. And they have exactly the same, with a large increase in the secretion of glucagon after these operations. And in fact, I think, you would think that this would be counterproductive because this would increase hepatic glucose production and be diabetogenic in a way. But in fact, I think it saves the patients from getting even more severe hypoglycemia. So in fact, I think it's a good thing.
Thank you.
Is that it? Does anybody else have any other questions or comments? Okay, so on behalf of the American Society for Metabolic and Bariatric Surgery, I would like to present this plaque to Dr. Holst to thank him for his presentation and our keynote speaker. Thank you. All right, so Dr. Holst is a quick speaker, and I thought we were pretty quick in the Northeast. So that actually will conclude our keynote presentation, our town hall, our presidential address. However, before I conclude everything, I do want to give a very special shout out and thanks to all the ASMBS staff. Without them, this conference is not possible, and a lot of the work that is done with integrated health, with the surgeons, we just wouldn't get it done. I do want to thank for the integrated health section, Jenny Wynn. Jenny, are you here still? Jenny Wynn and Mackenzie O'Leary, are you here as well? If you are, please stand up. No. Or else they're not going to stand up, which could be it. So thank you very much. I appreciate all that you do, and I appreciate everybody for coming to this. And again, fill out your evaluations and let us know what you need from us in the future. Thank you very much.